Hello, everyone, and thank you for joining us for day two of a virtual conference dedicated to practical applications of mitochondria research to human longevity and space medicine. Today is the final day of the virtual conference, and yesterday we focused on longevity space, and today's angle is space medicine. My name is Kate Butts. I will be a co-moderator of today's panel. I'm a managing partner of Deep Knowledge Group's longevity-focused fund, Longevity Capital, and I'm glad to be joined today by my colleague and co-host, Franco Cartiz, Director of Aging Analytics Agency.
Thank you, Kate, and thank you to everyone who's joined us for today's proceedings. Feel free to jump in the chat, say hi, let us know where you're tuning in from. And please feel free to pop any questions that you might have for our speakers and panelists in the Q&A box. We'll do our best to address those during the panel discussion later today. Practical applications of mitochondrial research for space medicine is the focus of day two, which has been organized for your viewing pleasure by Aging Analytics Agency. It's a broad topic, which we're very excited about, so much so that we're already planning a number of follow-up virtual and physical conferences on the topic of practical applications in mitochondrial research as it pertains to a number of domains near the end of this year and leading into 2022 in London and in the USA, as well as virtually, and further details on this in terms of specific dates, etc., should emerge in the coming weeks.
Today, we are delighted to be joined by a number of prominent speakers who will address the role mitochondria research plays in space medicine and protection of astronauts' health. Each panelist will deliver a brief talk, which will be followed by a joint Q&A session. This session is live, and we encourage our audience to pop up questions in the live chat. For those who could not join us today, this session is being recorded and we will distribute it thereafter.
And while the connection between today's discussions and day's one theme, practical applications of mitochondrial research for human longevity, it might seem slightly tenuous, in reality, we at Aging Analytics Agency and its parent consortium, Deep Knowledge Group, see many deep connections, synergies, and convergences between these two domains, in that many of the approaches to optimize the health of astronauts may also have a role to play in human health span optimization on Earth. And in fact, myself and no less than three of today's presenters, including Dr. Batut of the Belgian Nuclear Research Center, as well as Dr. Vaheshti of the NASA Ames Research Center, and Dmitry Kaminsky, general partner of Deep Knowledge Group. We're among the co-authors of a 2017 paper which touched upon many of these convergences.
Yes, and I think it's important to note that the human-centric approach to validation of safety practices and therapies, etc., is something that we hold very dear, and we hope that this approach will translate from space medicine to longevity industry space, because we all know that over the past few years, scientists have achieved great success in lifespan extension of model organisms. Unfortunately, however, the same cannot be said about human life extension. And space is one of those areas where, there's no other way other than to focus on human-centric approaches. And we always exhibited great interest in this area.
So before we kick things off, I think it makes sense to give everybody a taste of what we can expect from today's proceedings. First off, we'll be hearing from Dr. Afshin Vaheshti, bioinformatician and principal investigator at KBR, at NASA Ames Research Center, and he will be delivering a talk on mitochondrial dysfunction as a central driver of health risks associated with spaceflight.
Yes, and we'll then be joined by Dr. Sarah Batut. We are very happy to have her. Dr. Batut is the head of radiology unit at the Belgium Nuclear Research Center, and she is the recipient of the 2020 Woman of the Year Award by King and Queen of Belgium. And go
ahead. Following that, we'll be hearing from Kate Batts, who will be delivering a brief summary of an analytical report and IT platform on the femtech healthcare landscape in Q2 2021. produced by Deep Knowledge Group and Aging Analytics Agency's analytical subsidiary, MTech Analytics.
Yes, and we will then be joined by our very own Franca Curtis, who will deliver a talk on biomarkers of human longevity as the critical catalyst for longevity industrialization, governance, and practical applications.
Following that, we'll welcome to the floor Dr. Arkady Prokopov, biogerontologist, mitochondrial researcher, and physician for integrative medicine with over 3 decades of experience in normal baric intermittent hypoxic training and therapy, who will be speaking about whether or not mitochondria optimization can help human space exploration.
Yes, and our final speaker will be Dmitry Kaminsky, founding partner of Deep Knowledge Group. And Dmitry will deliver a talk dedicated to global space tech industry Q2 2021 overview.
And to close off today's event, we will be bringing together all of these speakers for a roundtable and panel discussion to delve into the various topics of today's presentations in a deeper and more dynamic manner. With all that being said, I would now, without further ado, like to welcome to the floor Dr. Ashim Maheshti to present his talk.
Thank you for the introduction, and thanks for inviting me for this nice event and session. All right, so I'll just dive right into it. As Franco and Kate nicely introduced everyone here, I had the pleasure of working with Franco on that paper that he was leading. We've actually never been in the same room with all these people, but now sort of in the same room now with some of the folks that were involved. But that was a nice experience and a nice paper people should read about like the general effects of space radiation and resistance. Frank already introduced who I am, but before I start talking about the actual meat of this, I'm going to dive, go really quick about some background. I'm sure everyone's familiar with these aspects of space flight, but one other aspect is resources that, what was used for this mitochondrial project, the data that people can have access to. So I'll probably go through this part really quick, so most people are really familiar with this already. So we know in the space environment, there's multiple hazards that are there. This right panel here is from a review paper we put out in Cell, the same time this other paper we're going to talk about last November. But this in that review, we had actually discussed, the hazards and fundamental features of space biology, and mitochondrial is one of the key ones we discussed in there too. But so the main hazards in space, most people are familiar with, is space radiation and microgravity. There's other ones here as we talk about confinement, distance from Earth, which I'm not going to cover. But as you can see in this little movie here, like one aspect of space radiation is solar flares from the sun. So solar particle events, otherwise known as a high acute dose of protons that astronauts might get exposed to in deep space, which doesn't happen all the time, but it can happen and be detrimental or potential increase of health risks, associated with that due to the damage caused by that solar flare. The other aspect is galactic cosmic rays. So as we know, all current experiments done in space are either done at the International Space Station or the formerly experiments done in the Space Shuttle before the International Space Station was done at low Earth orbit, which would be here. So low Earth orbit, as we know, is in the magnetosphere of the Earth. So then the dose that might be coming into, from the deep space radiation, the galactic cosmic rays is reduced. But a deep space mission to Mars or going to the moon, now you're exposed, you don't have that extra protection from the magnetosphere, and now you get the full-blown effects of all the, space radiation damage and the doses that you might get when you're going to deep space missions. This is like an older slide here from, because some of these terminology is outdated, but I like to show this still, but kind of gives you an idea, like on Earth, let's say if you get standard chest x-ray, PET scans, what the, accumulated dose might be per year. But when you compare it to like Apollo missions or the old shuttle missions, and then Skylab's supposed to be ISS, and then compare it to a mission to Mars when you go 1.5 years there and back. And then you also add the solar flare component that could occur once in a while. And you can see, overall, there's a lot more damaged or a lot more accumulated dose an astronaut will get compared to your average doses you get. And the other factor that you got to consider is on Earth, the radiation is different than what's in deep space. In deep space, you got the heavy ions, of course, like iron particles, a mix of iron particles, proton, oxygen, and so on, which is a lot more damaging than gamma rays or X-rays that you get on Earth. So, with all this, there's actually then, as you can imagine, a lot of concern for health risks, which a lot of them are associated with mitochondrial factors too, as I'm going to show you, which previously was, not as people knew about it, but, now more and more, I think people are realizing mitochondria is one of the key regulators. So again, you can look at all the different type of health risks associated with space radiation and with the microgravity component, so the lack of gravity contributing to the obvious things like muscle loss or bone generation, but that also contributes to things like SANS, which is space-associated neuroocular disease, which would reduce your vision, basically. And people think it could be because of fluid shifts in the brain or the eye, or the flattening of the edema and things like that that could cause the vision loss. But there's still a lot of unknown about it. It could be related to mitochondria, so that's the part of the research that there's a lot of unknowns that, in space, as we all know, that which kind of makes space biology type research, exciting, because what you discover sometimes is the first one. You'll be the first one to discover something. So just briefly, type of experiments that we do. I'm only focusing on the mice and the cell type experiments, but there's a lot of other organisms people work on, like plants, C. elegans, fruit flies, so on. There's a lot of literature out there on all these different organisms. But type of experiments that are done in space, one is you go to the International Space Station, you send organisms. This is a movie of our mighty mousternauts, as I call them. They're floating in space. And this is the cage they're set up, and the camera on the ISS, and you can see they're floating around, seemingly well adapted and adapting to the microgravity environment. So what happens is, people in payloads, they set up different things like cell cultures, or in this case, mice, and the astronauts will take care of what experiments that are there in space. So sometimes the mice are sent back to Earth, and sometimes the mice are sacrificed on the ISS. And then, if you want to simulate experiments on Earth, there's only a few places that you could do this. One place, which NASA had set up early 2000s, is at Brookhaven National Lab, which is a high energy physics collider facility, a DOE facility out in Long Island, New York. And NASA set up their NASA Space Radiation Laboratory in 2001 or 2002-ish, around there. And what it is basically, they set up this facility so you can actually radiate samples, whether it's mice or rats, or in cell culture experiments or anything else you want, in front of a beam, which is that would simulate space radiation. So you can do it like single ions, like iron particles, oxygen, silicon, so on, or you could do mixed beams now of different simulated galactic cosmic rays. So then this is one way to simulate space radiation experiments. And then for microgravity experiments, usually in rodents, these are kind of the main one is these hind limb experiments. You see the mice suspended by their tails. It's kind of silly, but that's the only way people could really kind of capture in vivo that unloading of the weight in the back of the legs and the muscles. So this experiment usually you do a hind limb suspension for a couple of weeks or a month or longer, however you want to experiment, and does a pretty good job of simulating that muscle loss that you might experience in space. There's newer models by Stuart Rudkov, a collaborator of mine, Martin True at Beth Israel and Harvard, where they're doing this partial weight bearing rat model where you could go up and down with the rat at different conditions to simulate different types of percentage of gravity, let's say on the moon or Mars or deep space and so on. So there's a different type of experiments. Now, where does, let's say you run all the experiments, you get omics on the experiments, where does it end up? Gene Lab is the place that it ends up. And I'm just really briefly going to go over it, but you guys could go here to genelab.nasa.gov and explore on your own. But Gene Lab is really briefly where basically all the omics related space biology end up. And it's free, open resource to the entire public, entire world. So if you have any hypothesis that you want to, tackle, it's free data. So you go there, mine the data, and play around with it. So what it has is a data repository, basically, where all the omics from proteomics, transcriptomics, epigenetics, and so on is on there. And then we also have portals that you could actually analyze data through this other platform. You can actually analyze your own data there if you're not a hardcore bioinformatician that you don't want to download data. And we also in Gene Lab have provided a high order data, like process data. So let's say if you don't want to do the processing yourself, you got the fold changes of genes or proteins and so on, and you can just download that and start, with the high order data and play around with that. And then also environmental data like accumulated dose that was experienced, let's say for samples that are flown into the space, and other things like collaborative workspace. And also we have a visualization portal. You could actually visualize the data. So you could actually just start there, maybe key mitochondrial gene that you want to look at, or protein, and see if it's there. And then you could evolve your hypothesis and questions from there. So basically that's where, this started here in Gene Lab, where we mined the free data here. And then one other thing is we have these analysis working groups in Gene Lab, which is here. We have 4 of them: animal, multiomics, plants, and microbe. And I lead the multiomics group here, systems biology multiomics group. And this was set up originally by Gene Lab to get scientists from all around the world, non-NASA folks, non-NASA PIs, to actually work on space biology questions, and also if we will have any pipeline, GeneLab has any pipeline things they want to consult the scientific community with, they can consult it. But what ends up is all these scientists end up, anyone can join, is free to join. And you get this large collaborative network of scientists working together on interesting space biology questions. And that was something I want to get into, but you also have an educational working group, not working work, but working group that, universities and schools are starting to work with to, use GeneLab as a learning tool. But the analysis working group was key to come up with this mitochondrial story I'm about to show you. And just to briefly background, these are the type of data sets we have from spaceflight experiments to ground experiments that you saw, and different types of omics and organisms. So how did we use GeneLab, and how did this story come about? So as I said, this is from a paper we put out in November in Cell. We got the cover. This is a picture Scott Kelly took from the ISS. So Cell gave us the cover, and Scott Kelly provided the nice little picture here. And this is a part of a larger package of papers that me and Chris Mason and Susan Bailey, Chris Mason's at Weill Cornell Medicine in New York, and Susan Bailey's at University of Colorado. And we approached Cell Press and put together this large package of papers with Cell Press, which is about 29 different space biology related papers, and they call it the biology spaceflight. You go to this link to see all the papers and read all about space biology. And Cell Press was great to work with to get this out. And this paper ended up in Cell. And what was basically, first, before I go on, this is, as I said, this is a work that was a large collaborative group work from all the analysis working group members, which you see here, and some others that, joined that analysis working, that the people from our group brought in. So, it couldn't really happen without a large collaborative network of people working together to do this kind of work. So basically what the idea is, we started this as sort of, it was a fishing expedition, which, you shouldn't use the science, but sometimes this is what you need to discover new things. So basically the question I started with to the group is there a master regulator switch that could determine that the body is regulated in space? And so the easiest and low-hanging fruit, of course, for all these AWG members is go to GeneLab and then look at the multiple data sets that were flown to space. So there's a lot of mouse data sets we looked at with all these multiple organs that you see here. There's all these in vitro data sets that we looked at, separate members looked at. And then we not only looked at the transcriptomic data, but we also looked at methylation data, proteomic data, and even metabolites, metabolomic data. And then did, all this higher order pathway analysis and modeling to see what happens. And what popped out is, over and over again, when people separately with an unbiased view analyze the data, and then we came back together and compared notes, mitochondrial functions, no matter what kind of omics you looked at, was the key top hit in the master regulator, no matter if you looked at any of these mouse organs or any of these in vitro cell culture experiments that they did on ISS. And what was interesting then, when we compared later through collaborators in JSC, Scott Smith and Brian Schwartz and Sarah Zor, that they have a lot of pathophysiological data from astronauts. So we gave them a list of things related to mitochondria that could be floating in the blood, or in your urine, in an astronaut urine. And then they compiled this, and this is the largest amount of astronaut data and be in one paper, but they compiled basically 59 astronauts from the past few decades into one paper, and then to confirm this. And then we also had the NASA twin study data on there, which I'm going to go into, which further confirms. So really quickly, since we're going to pack a lot of data at you, but you guys could go to the paper and read this too. So we started, with this paper, everyone was doing everything at once, but the in vitro data is this. So basically looked at these different cell culture experiments that were done on the ISS, and the data is available on GeneLab. And what, a long story short, is when you compare what pathways are in common for cell culture to be flown in space. And what popped out was all these mitochondrial related factors. Everything that's only in common. Sure, there's a lot of pathways that are not in common between these cell culture, but what's a common regulator that's occurring is mitochondrial function in cell culture alone in a dish, which was nice, and nice to see that this was a common theme no matter what you look at, or no matter what kind of cell type you look at. Now you go to the mouse tissue. So these are little complicated networks, but one of our members made this really nice complex network, but it's easy to look at also, where the outer ring of these network are related to the transcriptomic data. The inner ring are the proteomic data. So then now you can look at, like, this plot B here is looking at all these different tissues from liver, kidney, eye, and adrenal gland from the mice that were flown to the ISS. and also from two different sets of mice from two different strains. So there was a C57 Black 6 mice experiment that was sent to the ISS, and then there was a BALB-C one. And some of you guys familiar with the background of mice, and for the paper that Franco showed in the beginning, C57 Black 6 mice are more radio resistant and BALB-C mice are more radio sensitive. So there are key differences that happen when you look at the overall biology. But what popped out, no matter what you looked at the transcriptomic data or the proteomic data, no matter the genetic differences between the mouse strains, mitochondrial factors go in the same way. And then this is like the muscle tissue, which I'm going to get into in the slide next, the differences between the organ and the muscle and why it makes sense of mitochondrial activity. But again, proteomic data and transcriptomic data in these different muscle sets from quadricep to tibia to gastrocnemius muscle, and then the soleus muscle is here. We had a carotid artery, and EDL, the extender digitorum longus muscle. So, within all those muscle data sets, we saw again a theme occurring where I'm going to get into a few slides down where that indeed the mitochondria is actually suppressed in the muscle, while most of the organs, or not most, but some of the organs like the liver, the mitochondria is reversed. I'm going to get to that why. And this is just kind of another schematic, these heat maps here of of how the specific mitochondrial genes that was taken from, as a resource we all know, Mitocarta made by Broad, for the supposed active mitochondrial genes that are approved. So we map this onto the transcriptomic data, and one thing that this overall plot is a lot happening here, but one thing is that we see the liver is the one being most dysregulated in space due to mitochondrial activity. And there's other organs happening. And then when you look at the muscle, pictures of the way, you see that the soleus muscle is the other one the muscle that's being heavily regulated. And we know from experiments that the soleus muscle is usually the one impacted first in space. So that was interesting to see that. Oops, let me go back one. So now we're looking at other factors. So we looked at metabolites in the muscle activity. And once the analysis was ran, what we see in blue are pathways related to mitochondrial activity. And we see that what's key regulating, what's the top hits that are happening and a lot of universal things happening is mitochondrial pathways, directly related mitochondrial pathways in the metabolites that are being impacted by spaceflight. These are mice that were flown to space again. There's other activity like glucose, lactose, and glycolysis, which are not directly with mitochondria, but they are downstream of mitochondrial dysregulation, as we all know. So anything here like fatty acid metabolism, these are all pretty much related to mitochondrial, but the blue is the one things that are directly related. When you take apart like specific genes that are, like for example, oxidative phosphorylation genes or integrated stress response genes, we see key differences and key regulation happening in organs, which I won't get into the really nitty-gritty details because my time will be, I'll spend all day talking about this, but you could read the paper again where we discuss all the details and how the differences between the tissues and mitochondrial dysregulation. Now, when you compare it to the twin study data, so this is, if you're not familiar with this experiment, it's Scott Kelly was flown to, stayed on the ISS for a year. It was one of the longest stays, at the time when he was there. And then his identical twin, who was an astronaut, now he's a state senator, he was on ground for that year. And then basically, as most astronauts, when they go to space, they give all their samples to us to look at. And this is in collaboration with Chris Mason's lab. We saw that indeed, and Chris Mason's collaborators, this is looking at specific mitochondrial DNA nuclear markers. And you see that indeed the twin is here, that was in space, Scott Kelly, and Mark Kelly is the solid colors that is on ground. You can see there's not much changes happening over the entire year. And then there's pre-flight, was black, and then during flight, and then post-flight is the orange. But you see in Scott Kelly, these mitochondrial factors are being heavily upregulated when they're in space, which is not good. And then down here is the physiological data from blood, from different markers that we looked at from the astronauts, from the 59 astronauts over the past 3 decades, compiled together from the folks in JSC. So we see, for example, antioxidant markers are really like oxidation phosphorylation stuff is being suppressed. And then these markers from the urine, which are related again with cellular oxidative stress, are being heavily upregulated, which again is what you would expect if you get the mitochondrial dysfunction happening as we see in the mice. So the astronaut data is again showing that, confirming sort of the conserved nature between species from the mice to the astronauts. And then, what's interesting, let me go back one because I was going to say here, the metabolic flux analysis, this was done by Deanne Taylor's group in Children's Hospital of Philadelphia. which you take the transcriptomic data and then use modeling to actually predict metabolic flux activity that's happening. What's interesting here, this is for the liver, and these are all for the muscle tissues. You see that in the liver, mitochondrial activity is, and these are mice that were flown in space from the 2 different strains, and these are the ground mice, and these are, again, mice flown in space, and these are the ground control mice. So you can see that in the liver, the mitochondrial metabolic flux activity is highly up, but in the muscle, it's reversed. And, when you look at the cachexia literature, which talks about muscle degeneration, indeed, the liver tries to overcompensate for some of the issues that happen in other places, like your muscles. So this is indeed falls well with what people know about mitochondrial activity, and this is here. And what's interesting here, this part here is, again, the NASA twin study data, the transcriptomic data, and what we see as panel D. And this is tracking again, specific mitochondrial genes determined from mitochondria, overlapping with the transcriptomic data from Scott and Mark Kelly. And this is comparing the fold change values, where you can see basically pre-flight versus, Scott versus Mark Kelly, or comparing basically, Scott Kelly before and after flight. And the key take-home message from this is a lot of things you can look at here, but the key take-home message of this, when you look at these different comparisons, compared from different cell types that were looked at, is that over time, Scott Kelly's mitochondrial profile changes. So there's a shift that's happening. And you can see, for example, when you look at, let's say, in-flight versus pre-flight, or post-flight versus ground for, let's say, talking about comparing Scott and Mark Kelly, for these different cell types, you can see that, in some cell types, for example, mitochondrial activity shifts from, there's not much happening between them that's different. But then once he comes back, you can look at, for example, the CD19 population, or lymphocyte depleted population of cells, LD's lymphocyte depleted, is highly now different with mitochondrial activity, and so on. Or you can see a shift that's decreased in different, pre-flight versus ground. But this is kind of conditions that you see that, over time in Scott Kelly is being shifted. So mitochondrial activity is actually being shifted in a whole overall profile. I might be getting close to the end of my time, so I'll run through this really quick, but this is all downstream activity from mitochondrial that we know happens, for example, immune function and immune dysregulation or inflammation. Again, there's a lot of astronaut data that we have here. It's the astronaut data that you can see related with VEGF or inflammation or cholesterol levels that we see is heavily regulated. And the liver seems to be one of the key factors there. It's the methylation data, look at the immune profiles. This is interesting, fatty acid dysregulation having a lipid accumulation. Again, when you look at the liver compared to the muscle metabolic flux activity, that's just downstream of the mitochondrial. It's that inverse relationship that we see. And then cholesterol's here. Sorry, I said cholesterol's another slide, but it's here we see in the astronauts, again, cholesterol's heavily up, and some of it doesn't recover when they come back. And just to briefly end up here, we know that mitochondrial activity regulates, upstream of cell cycle or circadian rhythm issues, which are heavily regulated in the mice when they're up there. And then also what's interesting, one thing that popped up is like olfactory issues in the mice are being heavily regulated. And we know the genes associated with this when we look at the, pull apart the pathway analysis and look at what happened at the methylation. These genes are heavily related to mitochondrial activity again. So, and then there's olfactory issues similar, like, what we see with COVID, for example. Astronauts have reported, of course, sense of loss and sense of change of their smell and taste happening. And this pops up here in the mice. So overall, the summary of this work is a really simplified summary, is that indeed there's mitochondrial dysfunction happening when you're in space. And it seems to be like the universal driver upstream of what we previously thought maybe it could be circadian issues or lipid accumulation or anything else like immune suppression or immune inflammation that might happen in space. And I think mitochondria is above that, as probably the majority of people here would probably agree with. And so now potentially we could look at ways of mitigating this with potential mitochondrial therapeutics that people are testing in the clinic, and hopefully shift that towards astronauts and space biology work. And again, just a little plug, there's a lot of papers here, so take a look at that. Biology of Cell Flight, Cell Press. So you could not only look at this paper, but all the other papers that me and others, 29 other papers that people participated in. And then GeneLab, of course, is instrumental in this because they, provide the platform where we could mine it and do it. And hopefully people here might have other interesting mitochondria related questions that they could pop onto GeneLab and look at all the nice, interesting data where newer data sets pop up. So I think, 2 minutes over. So I think it's good. So I don't know if there's time for questions, but I'll stop sharing my screen.
Thanks so much for that, Afshin. There's a whole lot to unpack there, panel discussion. And with regards to Q&A, we'll save that, I think, for the panel discussion.
Sure, sounds good.
Yeah, but fantastic talk.
Thanks, Frank.
you.
Now we'd like to welcome to the floor Dr. Sarah Batut, head of the radiobiology unit at the Belgian Nuclear Research Center, as well as recipient of the 2020 title of Woman of the Year by the King and Queen of the Belgians, and recognized by Femme de Jourdois, excuse my pronunciation, as one of the 85 women that make Belgium move. And she will be speaking on the topic of personalized medicine for astronauts and radio protection in view of long duration deep space missions. Dr. Batut, it's our pleasure to be hosting you today. The floor is yours.
Welcome.
Thank you very much. Thank you. It's really a pleasure to be here, and thank you very much for organizing this workshop. I'm really pleased for the presentation that has been given by the first speaker, Afshin. That was really impressive, and I would like to now focus more on personalized medicine in astronauts. So as we know, we have now about 60 years of expertise, of know-how on what is the behavior of the body of an astronaut during a space flight and a space mission, and going and starting from the first orbit at Yuri Gagarin in 1961, an orbit of 1 hour and 40, to now on the ISS, on the International Space Station, we have expertise, and we know how the body behaves when these astronauts are staying for 10 days to 6 months to even more than 1 year sometimes. So we know that space flights have an important impact on the human body, and that it has some consequences, but these consequences really very much vary between astronauts, and we are going to see that more into detail. And what we are preparing, and it's an international endeavor, is the future, the fact that we're still using a lot the ISS, the International Space Station, but we are moving towards using and going forward to developing a moon village, going towards missions to Mars. So we are going to longer mission with higher risk. As mentioned by the first speaker, so we are in this endeavor, the deep space getaway, going to the moon, going to Mars, and this deep space getaway is the first human spaceship to explore the solar system. This will be the most distant human space mission ever attempted, and it's also a testing ground for challenges of long duration human missions in deep space. And one of the main recognized showstoppers is ionizing radiation. There is an international panel dealing with all the risks, the possible risks, potential risks for human space exploration. And this is the Multilateral Human Research Panel for Exploration. And as you can see here on this slide, this panel evaluates and ranks the different risks and rates the different risks the astronauts are going to be facing. And as you can see in the last column, this is a column where you see more red, and the red comes from the fact that the consequences of this risk can be quite high, and we see that radiation, which is the last lane on this slide, represents a very high risk, where it's absolutely needed to develop the necessary mitigation or countermeasures. So if we look at space radiation, as was already mentioned, we have three main characteristics and components of space radiation beyond low Earth orbit. First, we have the solar wind, which includes low energy electrons, protons, and alpha particles, which is a continuous flow, and which represents a low hazard. So it's not so much of a worry for us radiation biologists and radiation protectionists. But then we have the solar particle events, which are usually high energy protons. Usually it's an intermittent flux of these high energy protons, but it's very hazardous. So during that specific time of solar particle events, usually the astronauts are hiding and sheltering in very protected parts of the International Space Station. We'll see that in a few minutes. And then we have the galactic cosmic radiation, which are composed of mainly high energy protons, but also alpha particles, also heavy ions. And it's a continuous flux, hazardous, and for which we really need to develop further some countermeasures, some solutions. And we have also to take into account not only the first particles and the first flux of radiation, but also the secondary radiation. And these secondary radiation are due to the interactions of the primary radiation that I've just described with the nuclei that are present inside in the material of the spacecraft or inside the human body. So this secondary radiation can be even more detrimental to the human body than the first and the primary radiation. So it's also important to select carefully the material which is used for space flight, but also for a spacesuit, for example, to try to avoid at the maximum this secondary radiation production. Right now, the International Space Station is used, and is really important to understand and to develop mitigation ways and solutions in view of human space exploration to the moon and to Mars. So the ISS is really extremely crucial for all types of research, and we conduct a lot of research on the station. And right now we are conducting the immune follow-up of astronauts that have stayed at least 6 months to 1 year on the ISS, mainly Russian cosmonauts and European astronauts. We also have some detectors, some radiation detectors, because it's not that easy to characterize radiation in space, and we will see why in a few minutes. So we have detectors inside and outside the International Space Station. We also are very strongly involved in what we call in Europe the MELISA program, and the MELISA program is the life support, developing life support system, regenerative life support system in view of long-term missions. And we have sent 2 years ago the first bioreactor containing spirulina. So the level of radiation as we monitored on the ISS is roughly about 150 to 200 times the level of radiation on Earth. So the ISS is in orbit around the Earth 300 to 400 kilometers. So we are monitoring the radiation and characterizing the radiation through dosimetry purposes. And you see in orange in the picture some of the passive radiation monitors that we have on board of the ISS. And every 6 months we receive back these dosimeters, and we measure and characterize the doses, and we send some new dosimeters. We also have a mapping. We're also performing a mapping of the doses at different places. Here, as you can see in the Columbus module, where we can really measure, depending on the spacecraft material, on the coat of the material, we can really measure the dose that penetrates inside the ISS and inside the Columbus module, and that can differ from place to place. We also measure the doses that are received by the astronauts. I was mentioning to you that depending if you have a solar flare, you need a specific sheltering, and you see 3 places inside the ISS, inside the International Space Station, that are particularly protected, and where the astronauts can reach and stay for the necessary time in order to be well protected. And one of the middle module that you see there is the water wall, because water contains, of course, a lot of hydrogen. It's hydrogen and oxygen, but a lot of hydrogen that is very useful to block and to stop most of the radiation. So it's a place where astronauts will go and reach and protect themselves. The lab sleep station is also a well-protected area using polyethylene. Polyethylene is a long molecule of carbon and hydrogen on each side of the carbon atom, and also hydrogen is very well known to protect well. So that's also a place which is a good shelter. An astronaut is exposed to many different types of stresses. And we can distinguish between the physical stresses, microgravity, weightlessness, radiation, I mentioned already, and then the microbes. We have inside the gut the microbiome of the astronauts, and it's about 500 grams to 1 kilo of microbes. And these 500 different types of strains, of microbial strains, have a different behavior potentially in space conditions. Some will overgrow, will proliferate quite fast. Some others will inhibit their progression and their proliferation. So you have a dysfunctional, a dysbiosis, a change in the microbiome, which is very important to also quantify, characterize, and we are doing that also. We also have some psychological stresses that we have to take into account: isolation, confinement for a certain number of months, and also a very heavy workload that can induce also some different types of stress, and these sleep disturbances and circadian rhythm, which can be disturbed. And we can also, in a nutshell, as mentioned also by the first speaker, distinguish between the fact that you have space sickness, disorientation, a disturbed sense of equilibrium, also the movement, which is only recorded by the eyes, change of liquid distribution with a face thickening and thinner limbs. The kidney function is also increased, and calcium is also increased. Calcium certainly in the urine, that can induce a higher risk of developing kidney stones. Then the immune system can be disturbed, and we also have an increased microbial virulence that can take place, and loss of muscle mass and loss of bone mass. But this impact can vary between astronauts. And we also have, on top of this variability and variation between astronauts, we also have a difference between in the gender between female and male astronauts. And as we know, females have less hearing loss in space than their male equivalent. Females, also female astronauts, don't seem to have statistical increase of visual impairment, and they seem to have also a better immune response in comparison with male astronauts. But they seem to suffer more from orthostatic intolerance, and they also have more urinary tract infections. But again, between astronauts, you have also a large variability in muscle and bone loss, equal in females and in males. So a lot of variability. And when you look now to the variation in terms of cancer risks, it also, as you can see, a wide variation. When you look at the risk of cancer on the ISS, as you can see, you have 0.1 to 1%. So it's quite already quite an important variation between astronauts. But when it goes to going to the moon or to Mars, this variation increases even more. As we can see, we have an exponential increase for lunar colonies, with a risk which can be even up to 10% higher than on Earth, and then on Mars mission, even a risk that can go up to 40% higher risk. Then if you look at radiation sensitivity, we are not all equal in front of radiation. Some people can be very radiation sensitive. We see that in patients, and some people can be very radiation resistant. So if you look at the range of radiation sensitivity, there again, we see that through our studies on astronauts, that some astronauts are very radiation sensitive, whilst others are less radiation sensitive. So it's also important to know that there is also a wide variation and a wide variability between astronauts. Then radiation sensitivity is also depending on the age, with at birth, and certainly at the embryonic stage, fetal stage, a much higher radiation sensitivity than later in age. And that can also be important to take into account when you look at the age sensitivity for longer missions to the moon and to Mars. And I would like to show you also the table that you see on the left-hand side, where basically the risk above 50 years old become much less. So that can also be an important factor to take into account for longer mission and higher risk remission and higher exposure to radiation. So the dose of radiation, as shown here, you can go from Earth, you have about 1 millisievert on Earth, 1 millisievert per year, and you increase of about 150 times on the International Space Station. To the moon, we can roughly count on 300 to 400 millisieverts. So it's 300 to 400 times more than on Earth. And then to go to Mars and back, we are facing radiation doses that in total are about 1,000 to 1,300 times more. And as I mentioned, every mission is specific. So the radiation doses is also mission specific, because the destination varies, the duration varies, the vehicle and the habitat design with the secondary radiation that can vary, and also the solar conditions. So let's speak a little bit about the moon and the International Moon Village, which is really an international endeavor in which humans and robots will work together on mining operations, astronomy, and or other projects. And the moon is very important as an archive of the Earth, her early history, but there is no magnetosphere, there is no general atmosphere, so there is no real protection from harmful space radiation. But it will be the moon experiments, and the moon station will be the springboard and testing ground for future exploration to Mars. We also have to, for the moon, we also have to design and rethink very deeply the spacesuit, because what we know from the Apollo program, there were less than 20 extravehicular activities during the entire Apollo program on the surface of the moon. But the future lunar programs are requiring a much higher level and doses and level of extravehicular activities for exploration on the surface of the moon. And we reckon of about 75 extravehicular activities per astronaut during their 6 months mission on the ground, on the moon ground. So this spacesuit will be the only protection during these exploration missions. That needs to be, this spacesuit needs to be resistant to abrasion, to reactive chemical dust, to UV temperature, oxygen, and weight. Needs to be suitable for walking, so it needs to be flexible, elastic. So it's very important also to develop the necessary know-how and expertise on the best spacesuit to protect, especially from radiation. And then on Mars, we are counting of about 1,000 times the dose in terms of the whole mission to go and back, being exposed to the deep space radiation and back. The Mars planet is the most similar planet to the Earth. It will allow us to understand whether life existed elsewhere in the universe beyond Earth. It has been once full of water and warmer, and it contains a little bit of atmosphere, 1% atmosphere. So it's potentially an habitable environment. What are the space radiation health risks? Well, first, we have a higher risk of developing cancer, radiation carcinogenesis, but also higher risk of developing acute and late central nervous system effects, like effects on the cognition, on the motor function, on the behavior, on the mood, and on the neurological disorders. Also higher risk of developing cardiovascular diseases, stroke, but also cataract, and also diseases related to aging. And then higher risk of developing, due to solar particle events, of developing acute radiation syndrome, which is including symptoms like nausea, vomiting, anorexia, fatigue, skin reddening, depletion of the bone marrow and of the blood forming organs. So we are really moving also forward to personalized medicine. So from the current approach, astronaut approach, which is basic, same countermeasures for everyone, for all the astronauts, we are moving towards a personalized medicine approach for astronauts, which takes into account their sensitivity, that takes into account high level data at the level of phenomics, of genomics, transcriptomics, exposomics, and metabolomics, and proteomics, and as well as epigenomics and microbiomics. So all these data are very important to gather before astronauts are going to space, to know them, to know the basic values, and also to be able to follow that up and to use a countermeasure when needed during the mission. And what is for to develop this personalized medicine, you need also the proper biomarkers. And what is a biomarker? Biomarker is any measurement reflecting an interaction between a biological system and an environmental agent, like the space environment. And that can be chemical, physical, or biological. There are different types of biomarkers, and especially for radiation, you have the biomarkers of exposure that can give you a direct idea of the level of exposure and the dose the astronauts have been exposed to. Biomarkers of susceptibility, these are genetic biomarkers that can give us an idea beforehand of which astronauts might be more sensitive to the space environment. Then you have biomarkers of late effects, so being able to predict in advance what type of reactions the astronauts might encounter. And then biomarkers of persistent effect that starts to develop from the moment of the exposure. And so if you look at these different categories, you can see that you have some cytogenetics biomarkers, mutations, transcriptional and translational changes, as well as DNA damage changes. So my institute, as the Belgian Nuclear Research Center, is working on different biomarkers. Here is a beautiful articles that have been published a few years ago with wonderful scientists, and most of them are here around the table and are presenting today. So, and this article is Vive la Radio-Résistance, which is especially devoted to what are the different ways that we can improve radiation resistance in astronauts. And that's going to be very useful and very important to tackle each of these ways, as you can see here, enhanced shielding, medical selection of radiation resistant individual, radioprotectors, gene therapy, biomarkers, hibernation, and biobanking, and regenerative medicine. So all these are very important issues that need to be tackled separately or together in order to make sure that we have safe trips to the moon and to Mars. And just a few slides about what we are currently doing at the Belgian Nuclear Research Center. We are working on different ground-based simulation platform, the random positioning machines, the bed rest studies, also different isolation platform in Antarctica. I was myself in Antarctica, and we're also flying different experiments that will show. So we are monitoring the individual radiation sensitivity of astronauts in space. We have spent 6 months to 1 year in space. We're also studying how the immune system and how isolation and confinement is changing metabolism and is changing basically how stress is changing health in different volunteers. And I was very happy myself to go to the Princess Elizabeth Station in Antarctica, but we are working mainly on the Concordia platform in Antarctica. We're also using a lot the bed rest study model from ESA, studying what is the impact of simulated microgravity on human health. And we're also irradiating a lot, so performing irradiation campaigns at GSI, but also in space, as you can see here, a skin experiment that flew in space a few years ago. We also had some cardiovascular experiment that flew to the ISS with the SpaceX program. And we also studied how the brain, how neurons are formed, and how the plasticity of the brain can be changed in space conditions. Important also to develop life on chip models for space in order to regenerate different types of tissues when needed, and to also keep a healthy gut microbiome. Very important to understand and characterize what are the most important bacteria that play a most important role that we need to keep and maintain in space conditions. And we flew also twice some rotifers in space. The second experiment came back 2 months ago, where we could really understand what are the molecular mechanisms that have been developed by these rotifers in order to endure and to be resistant towards radiation. So in conclusion, it's very important to develop this better personalized risk assessment for deep space exploration. We need more insights into the possible interaction effects of space flight stressors, if they are synergistic, if they are antagonistic or additive, and in order to reveal altered cellular functioning and as indication for potential countermeasure development. And let's not forget that all this space research is very useful on Earth in order to understand and to develop all different types of medical therapies, amongst others, better radiation therapy for cancer treatment. So I would like to thank you very much for your attention, and I'm open for any questions. Thank you.
Thank you very much, Sarah, for this excellent talk. Exciting stuff indeed. You've mentioned biomarkers as part of your presentation, and it so happens that our next presenter, Franco Cortese, will further develop this theme and address the human biomarkers of longevity and the critical role they can play in longevity industrialization, governance, and practical applications. Franco, welcome.
Thanks so much, Kate, and thank you for that very stimulating presentation, Sarah. I can't wait to unpack some of that in the panel discussion later today. Yes, my name is Franco Cortese, Director of Aging Analytics Agency, which is Deep Knowledge Group's longevity-focused analytical subsidiary. And I will be delivering a short overview of a new analytical report and IT platform, open access, which we've produced on the current state of the global biomarkers of human aging and longevity ecosystem, while briefly touching upon the role that biomarkers of human aging and longevity can play within the field of space and space medicine. So Aging Analytics Agency in 2019 produced an analytical report which presented at the time an overview of the current state of the field of biomarkers of human aging and longevity, which we produced as part of our parent consortium's ongoing prioritization of this domain of research and development in a number of specific strategic domains. But first and foremost, as the most readily available means for scientists, companies, and other institutions to begin switching to a more human-centered approach to validating various methods of human health span extension and practical longevity. Given the rapid pace of development in this arena since then, we decided to release an expanded and updated version of that analytical report, current up to Q2 2021, which we have produced for Longevity.International, a nonprofit international decentralized longevity industry knowledge and collaboration platform. Alongside the report, we also built an associated interactive IT platform, which allows the report's content to be viewed and interacted with a greater degree of personalization and precision. And which allows many of the individual biomarkers, biomarker panels, as well as the institutions supporting and developing them, including companies, R&D hubs, and investors, to be analyzed and reviewed at a deeper, finer-grained level. The full scope of the report and IT platform aim to illustrate the current state of the global industry and academic ecosystem of biomarkers of aging and human longevity, presenting categorized lists of 150 single biomarkers, as well as 100 diverse biomarker panels, many of which are market ready and approved for clinical use today, which are also ranked according to their ratios of accuracy, availability, and actionability or scalability, according to the notion that in many instances, the perfect is the enemy of the good, and that the use of biomarkers of human longevity that are good enough in terms of accuracy, but have a much higher degree of availability, implementability, and scalability, are in many cases preferred over the use of biomarkers that are highly accurate, but expensive, invasive, and therefore less scalable. And a major take home from that report is that the scope of individual biomarkers and biomarker panels, as well as the scope of companies developing these biomarkers available in a usable, scalable, market-ready, and technology-ready form for the use by scientists, companies, investors, and even governments in order to validate the effect of various therapies and approaches on human health span extension is indeed quite large, and in fact, probably larger than many viewers actually realize in practice. And more than sufficient to allow them to begin being used more comprehensively within the longevity industry as a tool, again, for human-centered, safe validation of different approaches to health span extension. We've seen a large diversity of opinions in terms of the role that biomarkers of human aging and longevity have to play within the broader industry landscape and feel that the actual number of market-ready biomarkers available for use, as well as their role as essentially the front line for leapfrogging the translational gap between model organisms and humans, is frankly not as commonly recognized among industry players and participants as it should be. A great example of this would be blood-based biomarkers of aging and longevity, or so-called hematological aging clocks. Now, I'm a bit biased, having co-authored a few scientific publications on the topic of hematological aging clocks. But the point remains that blood-based biomarkers, for example, especially when coupled with AI and deep learning-based components, and analysis have respectable levels of accuracy. One, which we publish in scientific reports, predict age with a mean absolute error of just 5.7 years, for example, which is not the best, but fairly good, at least compared with higher accuracy types of clocks like transcriptomic and methylomic aging clocks. But critically, and most importantly, blood-based biomarkers of aging and longevity and hematological aging clocks use biomarkers often from routine blood work. So in other words, extremely non-invasive biomarkers available to the majority of clinicians and even family physicians and doctors. So this makes them extremely scalable, much more so than more expensive or invasive variants like, again, transcriptomic or methylomic clocks. And often lesser degrees of accuracy is a fair trade-off in terms of gaining scalability, especially for specific stakeholders like governments, who we at Aging Analytics Agency have argued many times over the past several years should really be utilizing to a greater extent anonymized population level biomarker data to track the effectiveness of their efforts to increase national levels of health adjusted life expectancy and quality adjusted life years. As the United Kingdom is trying to do, in fact, via their healthy aging industrial strategy and their commitment to add 5 extra years of socially inclusive healthy longevity to their population by the year 2035. The central motivation of this report and platform is about more than just gaining deeper insight into a specific sector of the longevity industry. It's really about showcasing the fact that the capacity for industry players and stakeholders to begin transitioning to a more relevant human-centered framework for validation, as well as for refining the effectiveness of approaches for human health span extension, whether we're talking population level or individual level, and whether we're talking about specific therapeutic modalities, lifestyle and behavioral interventions, or even social policy measures in the case of longevity governance, is greater than ever before. And of course, this is all compounded by the fact that we're living in a digital age with mounting volumes of digital biomarkers, as well as in an age where the scope and power of artificial intelligence is rapidly increasing, which creates a perfect storm for synergy in that more data often results in better trained models, and the fact that increasingly robust forms of AI are in fact not just complementary, but in many cases fundamental to make sense of these ever mounting volumes of data in the first place. Much of this emphasis on safe human-centered approaches and the safe acceleration of R&D timelines should all be quite familiar to our space medicine-focused presenters today, in that the space industry and the subdomain of space medicine have for many decades served as an excellent example of a sector which prioritizes a safe human-centered approach. Meanwhile, the actual synergies and convergences between practical longevity and space medicine should also be quite apparent to our panelists and presenters today, both in the fact that various biological and physiologically detrimental effects of spaceflight share commonalities with the process of aging and have detrimental impact on many of the hallmarks of aging in particular. And likewise, the fact that many approaches to optimize astronaut health may also yield effects in terms of human health span extension on Earth, and vice versa. As we and various colleagues, including Ashim and Sarah, argued to be the case in our 2017 paper, Vive la radioresistance, converging research in radiobiology and biogerontology to enhance human radioresistance for deep space exploration and colonization. And given the title of today's conference, I would be remiss not to reiterate Afshin's earlier point that mitochondria are indeed a key driver of health issues in space flight, as again illustrated by the paper and indeed the pack of papers which they published in Cell late last year. And again, as Afshin mentioned, further validated by the NASA Twins study, landmark year-long study that compared the effects of spaceflight on a wide range of biomarkers between twins, one spacefaring, and alas, one still earthbound. Which in some, I believe, paints a convincing case for the fact that we can expect to see, and indeed should push to promote to a certain extent, greater levels of synergy and coordination between attempts to optimize human health span and practical longevity on the one hand, and the health of astronauts in space, which is a topic that I hope we can explore in much greater detail during our panel discussion later today.
Great, thank you, Franco, for your presentation.
And
I believe I'm next. I will start by saying that I would like to provide a brief overview of our FemTech healthcare report, which was recently released. It is not specifically focused on space medicine. However, as Sarah rightly noted, the health of female astronauts is definitely needs a specific angle to be addressed. So we hope that FemTech analytics in general will receive more attention, and the sector as well, but also that the same will be true for female astronauts. So I would like to offer a brief overview of our recent analytical report, which is in fact is a follow-up for our analytics that we released in 2019. FemTech is relatively young, but actually a rapidly growing space. And our belief is that it will become one of the major disruptors. By FemTech, we mean female reproductive health, we mean female longevity, mental health, and related areas. As always with our reports, and this is an open access analytical report, which we invite everyone to download on agmanalytics.com, we introduce a framework or classification for this space. So we've segmented the product, services, etc., by application, by product type, by stage, by customer. It is very clear that reproductive health, followed by pregnancy and nursing, as well as beauty, are a few of the dominating sectors here. In terms of frameworks by application, again, the leading areas are fertility, pregnancy, longevity, beauty, diagnostics, pregnancy is accompanied by an interactive IT platform, as many of our are, and it features over 1,500 companies, over 1,000 investors, R&D centers. This platform is interactive, so you can mix and match and see which investors invest in which space. So I invite everyone to explore it. In terms of FemTech market size, it is currently at 21.7 billion, and it's expected to grow to 6.1 billion by 2027. We actually believe that while it's an impressive growth rate, that this market sector is underestimated and is bound to become a market disruptor in the future. One of the important factors here is that one of the reasons why this sector is emerging is the fact that there is a shift in societal perception of female health. This chart actually is quite fun because it represents life expectancy of men and women in different countries. And while the overall trend is always that women live longer than men, but the difference actually is quite significant sometimes depending on the country. For instance, in Russia, women live 10 years longer than men, which is quite remarkable. In terms of regional distribution of longevity companies, North America leads, followed by Europe, Middle East, and Asia. In terms of revenue overview, most of the companies are in 1 million, 10 million range. Funding-wise, most companies received 1 to 10 million, and the chart on the right represents the top 10 companies receiving the funding amounts. Speaking of sectors of femtech, the leading one is pregnancy and nursing, reproductive health, pregnancy and nursing, followed by longevity and healthcare. Again, regional distribution, US leads, followed by Europe. Speaking of product overview, the main focus is actually on services and combinations such as device and service, app and service, femcare product, and farm product. And it is interesting to mention that actually companies that offer products, they generally only offer one product predominantly. In terms of funding per sector of femtech, healthcare, which includes general healthcare, cancer treatment, and sexual health, dominates, followed by reproductive health, longevity, diagnostics, pregnancy and nursing, and beauty. And this is an example of femtech companies by estimated revenue. The ones in the top quadrants are the ones focusing on diagnostics. In terms of publicly traded femtech companies, they're not that many. I believe there are only 3% that are publicly traded. Most of them are based in US. And this is an example of a visual example of how femtech publicly traded companies feature. In terms of using AI, we believe that about 6% of femtech companies are utilizing it. And the applications involve diagnostics, personal care and well-being, pregnancy and nursing, reproductive health, beauty for femtech, and healthcare. This is an illustration of top femtech companies using AI. And in terms of R&D centers and their regional distribution, North America leads again, with US leading, relatively closely followed by Europe in this case, and then Asia. In terms of investment landscape overview, we see the trend is exhibited here. Again, we believe that there will be an upward trend in terms of investing in this space. And because a few traditional investment funds are not solely focusing on femtech, there are actually a few that do. And coincidentally, these funds are usually comprised of women, and portfolio is one of the examples. So if anyone in the femtech space is looking for investors, these are the good examples here. Femtech investors distribution by region and funding, North America leads, followed by Europe and Asia. In terms of investors by sector, we have reproductive health leading the chart, followed by healthcare, longevity, pregnancy and nursing, and beauty. The key market trends in this space are personalization, endometriosis, in-home testing, menopause, fertilization, AI for femtech, tracking apps, and wearable devices. Menopause solution is actually one of the prevailing trends here, and of course the Asian population plays a role here as well. Reproductive health segment obviously is projected to expand significantly by 2026, and will be estimated to be at 41 billion. Tracking apps also play an important role, and we see lots of activity in this space as well. And in terms of key market development factors and future focus in femtech, the most important factors we see here is accessibility, favorable regulation, B2B partnerships, customization, and affordability. This is, a more granular look at our interactive dashboarding platform, which give you further insights on everything to do with companies, investors, R&D centers, influencers, etc. I do not want to delve in too deep on it during my brief presentation, but I encourage you to take a look. So with that, I will revert back to Franco, and I hope the report is downloaded. It is an open access.
Thank you so much for that, Kate. Very interesting presentation as always, and I very much look forward to discussing some of the convergences to be had between femtech and space tech and space medicine, especially as it pertains to reproduction and reproductive longevity off of Earth. So thank you for that, Kate. I would now like to welcome to the floor Dr. Arkady Prokopov, biogerontologist, mitochondrial researcher, and physician for integrative medicine, with 3 decades of experience in normobaric intermittent hypoxic training and therapy, who will be delivering a talk on whether mitochondria optimization can help human space exploration. Delighted to have you, Arkady, again, and the floor is yours.
Thank you, Marco. So first of all, I must confess that I was not that much involved in human research with cosmonauts because it was parallel. When I was working in Institute of Biomedical Problems, I was more concentrated on saturation diving. But because both environments are physiologically very close on the physiological and pathophysiological effects that they cause in humans, it was many cross, so to say, interactions. And also I remember that American astronauts who experienced both flights, orbital flights, and saturation diving experiments, they confessed that saturation diving is much more stressful because confinement is much smaller and the environment is much more aggressive. So in any case, it was a good model of continuous stress, and the factors which exist in saturation diving confinement, they are very close to space flight, orbital space flights. This article, this publication as a landmark was, I show it the 3rd time here, I think, in this presentation, because it's really a seminal article which systematized the functions of mitochondria and the importance of mitochondria in space flights, in space biology and space medicine. But I need to focus on some specific things which just shed some light on specific problems. For instance, the reaction of brain, the degradation, we would say, of grain matter, so neurons, and also white matter presents a serious challenge in space flights. And also it relates to sarcopenia and osteoporosis. All these effects, all these mechanisms, they have actually the same molecular biological background. It happens because mitochondria degrade much quickly in hypogravitation and under influence of cosmic radiation. And the replenishment of tissues from stem cells is retarded anyhow. And we see the slow degradation, accelerated degradation of tissues because of also not sufficient replenishment from stem cells. And of course, mitochondrial degeneration. In relation to neurons, we need to remember that the mitochondrial biogenesis takes place in the near of the nucleus, and also mitochondrial degradation, mitophagy. But with the neuron, we have very specific situation. Mitochondria shall be transported through the axons to the synapses, and about 60% of volume of synapse are mitochondria. It means that mitochondrial biogenesis should be very intensive, and mitochondrial trafficking should be also very intensive, because in axons, mitochondria undergo accelerated degradation, especially under condition of stress, whether it's physical stress or operational stress, for instance, like operators under very long missions, especially under stressful missions. So we have in such cases the very serious problem, accumulation of degraded dysfunctional mitochondria, but they cannot be utilized or recycled here. They shall be transported back. And this transport takes time. For instance, 2 millimeters a day, the speed of transportation of mitochondria. Now multiply it on the length of axon, and we get very impressive numbers. How does it look? This video shows transport of mitochondria, mitotrafficking in mice neurons. It's accelerated, of course, time loop, but we see that transportation goes in both sides, yeah, retrograde and anterograde. And this process is extremely vulnerable to various damaging factors. Most of them, of course, the most important is the radiation and problems with capillary blood, because white matter is very poor capillarized. Therefore, this is, so to say, locus minoris resistentiae. It was known long ago that using hypoxia for prevention of radiation damage can help in treatment, in radiation treatment of cancer patients. For instance, the first patent on this topic was issued in Russia at the end of 70s. And since then, this method was used in many European clinics to increase the protection of healthy tissues during radiation treatment. Here we see in this article that mice, this experiment with mice were breathing 10 to 8% oxygen during irradiation, and oxygen saturation decreased respectively, and hypoxia mitigated the lethal effect of gamma rays and improved the recovery of hematopoiesis in compartments of pluripotent and committed progenitor cells. Enhancement of the proliferative activity in hematopoietic tissues by a cytokine or immunomodulator dextran sulfide increased the effect of hypoxic radioprotection, while elimination of proliferative cells by hydroxyurea decreased the radioprotective effect. And clinical use of this phenomenon is actually, it was used pretty intensively. I don't know how it's now, because of course there are always some new pharmacological approaches and some new ideas, but this natural method I think should be not forgotten, and it has, of course, relation to radioprotection in space. In this publication, they treated women in tumors in abdominal area, and the results, despite increase of dose at large field irradiation in most cases, no difference were found between group and A concerning acute or late effects of radiation therapy. But conclusion, the aerogen acute hypoxia radiotherapy is so far not only the simplest, but also one of the most effective selective radioprotective methods. The nature of oxygen effect is well known. Higher oxygen partial pressure synergized with radiolysis under irradiation, and water radiolysis is always takes place. Amount of free radicals increases under higher oxygen pressure, and therefore the total radiation damage is much higher. Here we see the difference between hypoxia, these are survival fraction of the cells. Under higher oxygen, it's much less cells survive, and under hypoxic influence, cellular survival is much better. Here we can see that even small decrease of oxygen partial pressure, for instance, here in the air we have about 160 torr, but small decrease of oxygen partial pressure, and this decrease doesn't influence significantly our physiology. So it's in the window of hypoxic tolerance, but it significantly decreases radiosensitivity. And this can be obviously used for radioprotection. Also, of course, we cannot see in details radioprotection from different particles, from different parts of spectrum of radiation, cosmic radiation, solar radiation. So, of course, it should be studied further. In hypoxia, of course, effect of mitochondrial hormesis is very prominent, is very important. Under moderate hypoxia, we see the hormetic response, and it increases, mortality increases linearly under ROS exposure, but during hormetic exposure, during hormetic response, under hypoxic influence, it's much better tolerance. And interesting also that exogenous antioxidants, they eliminate effect of hormesis. So I often see that many patients are so lured into the antioxidative supplementation. They take a lot of antioxidants, and that doesn't help to improve their health. And the explanation is here, because hormetic effect need controlled release of free oxygen radicals. If this release will be blocked by antioxidants, we don't see hormetic response, and we can see only continuous degradation. Among substances and interventions that influence mitochondrial hormesis, hypoxia takes very important part, very important role. Of course, it's a synergistic action from many components, but hypoxia integrates practically all of these pathways. In 2001, I was presenting on this topic on the application of benefits of artificial and breathing environment for space habitats and space flights. And it was one of the first conferences on this topic. Many aspects were discussed. For instance, also application of hypoxic hypercapnia. Physiological hypercapnia also plays very important role as a very powerful protection mechanism against peroxynitrite. And later, these applications now are confirmed in many studies. Here we see, for instance, application of hypoxic gas media for specialists working in hazardous occupations. The program based on the use of normobaric hypoxic media can be considered as a selection method for the physiological support of emergency restoration of health of specialists in hazardous occupations. The other thing, the direct effect on psychophysiological training in submariners. It's also similarly to space flights, confinement is of course much more comfortable, so to say, but nevertheless, the elements of typical stress of staying in confinement for a long time, it's also found here. And using of hypoxic environment shown to improve operator capabilities of submariners. I must say that we use this approach also for flight control operators, which are known to experience very serious psycho-emotional stress, and the application of hypoxic training helped immensely in such cases. And there are specific effects revealed on the human brain protection from the damaging effects of hypoxia. So prolonged hypoxia definitely cause a very serious damaging effect, but if it is if the preparation is given with intermittent hypoxic training, the resistance of brain increases very significantly. I think that this is also important not only for people working in stressful environments, but also for managers, for people who work under stress conditions, irrespectively of is it a special environment or just stress from their professional occupation. Very interesting aspect of application of artificial breathing atmosphere is the fire protective action of the low oxygen, normobaric low oxygen environment. This was first noted in the beginning of 60s with the first orbital flights. It was noted that normobaric hypoxic environment can prevent incineration. And later it was conducted in many experiments. I was also participating in such experiments when working in New York. And also on this conference on Mars Society, we also presented a fire safety system, which was based on the phenomenon of ignition suppression and flame extinction in normobaric hypoxic environment. How does it look? You can see here. This is the showroom which we built in New York in our office. And these gentlemen, they stay in hypoxia. Also, they don't know about this. The environment here is about 13% of oxygen, normobaric oxygen, instead of 21. And as you can see, just taking any burning substance in this environment eliminates fire completely and instantly. So for human occupied facilities, this solution is an absolutely the best solution. Yeah, nothing can be compared. No toxic chemicals, no residuals. Here you see the ball with room air, yeah, taken in the hypoxic, normobaric hypoxic environment, and fire immediately eliminated by just opening this ball. And the gentleman tries to set the fire on the match, but it doesn't work. Interesting that these gentlemen who are here, two of them have metabolic syndrome, high blood pressure, and one of them has 3 or 4 stents after heart infarction, but they even didn't feel that they stay in hypoxic environment. So it's one more demonstration how safe this environment can be even for people who are not perfectly healthy. Such environments are used now in special facilities where the evacuation of people is not possible. It has application in military, in special operation control facilities, and the safety of such approach is confirmed many times. And now some practical things. Recently, I started to consult a patient, a boy who is 6 years old, with the diagnosis of Leigh syndrome. He has homozygote mutation of SERF1 gene, and it causes deficiency of cytochrome C, COX deficiency, this is complex 4 of mitochondria. And the boy has retardation of motor function and mental function. So he was practically hard to move. He was staying in the bed and hardly could do anything. So we started to look what could be done to alleviate his condition, because there is no question of treatment with such syndrome, but some approaches are possible. And the idea was on the treating mitochondrial disease with hypoxia. This article was published in, so I think about, in 2016, and it was research with the mice, model mice who were treated with 11% continuous hypoxia. And in this environment, in this atmosphere, they stayed healthy. They stayed completely compensated, and even their aging tempo was not accelerated. So why not? We decided to try this. This is this article. This is a model animals which were rescued by intervention which normalized brain hyperoxia. The problem is that even under normoxic conditions, their brain is in a situation of hyperoxia, relative hyperoxia. And of course, it has multiple negative effects. But when they were placed in an environment, normobaric hypoxic environment with 11% of oxygen, everything was compensated. So we applied hypoxic tent, which is ventilated with hypoxic air with the concentration of 14% oxygen. And the boy, after 3 weeks sleeping in this tent, he started to move. He started to be more active. Started speaking more actively. So that was very interesting. Now, and you see the oxygen saturation is 91%. And on the pulse oximeter. Now we try to reduce. We try to reduce environment to, let's say, 12%, because he need to stay in the tent at least 12 to 14 hours a day. But this is a chance for boy to continue developing and to improve quality of life. I must say that in relation to space research, to space explorations, I would say that there will be a special time when decisions should be made. People should be genetically modified to endure long-term space flights and also staying in planetary habitats, because we know that it's virtually impossible to completely protect from irradiation, and oxygen will be always a problem. It will be always a narrow point, so to say. So I would, if I were this boy, if I knew that I can flourish in the atmosphere of 11% of oxygen, I would sign myself for the space flight where hypoxic environment is employed and where I could live relatively normal life. That's my opinion, of course. I don't say that it's, I so to say, impose this idea to anyone, but I think that future applications and the future challenges, they need artificial environments on the one hand. On the other hand, people should also adjust to new environments and new challenges of space exploration. Thank you very much for your attention.
Thank you very much, Arkady, for this helpful overview. I indeed learned quite a few fascinating facts, as I'm sure did the audience. It is now my pleasure to welcome Dmitry Kaminsky, a founding partner of Deep Knowledge Group, who will cover the current state of space tech industry. Dmitry, welcome.
Yes, hello everybody. It's my great pleasure to be here today, and I really, with great attention, was listening to every panel, to every presenter today, because indeed information is extremely interesting, and I do see even more now ideas about how space medicine is actually interconnected with human longevity on Earth. So, as I said, today I will provide some considerations how space tech medicine is actually interconnected with longevity industry and practical human longevity on Earth planet. In our consortium, Deep Knowledge Group, we have 10 analytical subsidiaries, including Aging Analytics Agency, presented by Frank Curtis. So this company was established back in 2013, and this company is focused apparently on aging research and longevity industry, and also practical applications of anti-aging techniques for extension of healthy period of life. We also recently launched Space Tech Analytics. This company apparently focused on space tech industry. We have a number of other analytical subsidiaries, including Femtech Analytics, represented by Kate Buzz, Deep Pharma Interns, Neurotech Analytics. So half of our analytical subsidiaries, they somehow related with healthcare, with advanced biomedicine. So as I mentioned, Space Tech Analytics produced this special case study, Space Tech Industry 2021 Q2, quarter two. This is the mind map of the industry. So far, there are 10,000 companies in this industry, 5,000 investors, and several hundreds of hubs, R&D hubs, associations, government organizations. So, and one particular sector which Space Tech Analytics profiled is space medicine. It's not that big compared with entire industry, but nevertheless, it's quite visible. Again, this is distribution by companies, distribution by quantity of companies per each sector. Again, space medicine is not big, but nevertheless, there are at least 48 companies specifically focused on space medicine. Here's the mind map for different type of organizations, international organizations, government organizations, associations, and so on. This is regional distribution of companies. For example, US have 52 persons on a global scale. And this is how each of the countries having their position in global space race considered by quantity of satellites launched. Here's the budget. So again, United States quite far ahead compared with other countries, but nevertheless, other countries, many of them have quite significant budgets. Space Tech Analytics also launched specialized big data analytical dashboards. Currently, they're analyzing close to 1.5 million data points. So this is big data analytics. You can visit this dashboard by this link. There's quite a lot of information. At the same time, this is first iteration. So this dashboard, during this year, it will be enhanced through several iterations. And there will be quite more information, quite additional information, including there will be more and more information, particularly on the topic of space medicine. Here are several leading companies specifically focused on space medicine. And some of them actually not only in the United States, but also in Canada, in UK, in Europe. Here are the organizations, including universities, which are dealing with space medicine. For us in particular, the most interesting part of this entire field, it's biomarkers. Today, during presentations of other prominent speakers, in particular scientists, they several times mentioned biomarkers. So indeed, markers are crucial for any industry. In case of medicine, biomarkers, like actual human biomarkers, this is the entire, the most essential, the most crucial bottleneck for actual understanding how to proceed further in a tangible, practically applicable, human-centered way. This is, of course, very relevant for space medicine, but for human longevity on Earth, it is even more relevant, just because we have yet an issue with genetic research that there's too much successful experiments on model organisms, including mice, but not enough in terms of actual humans. This article was mentioned many times today, so it's an honor to me that I was also among one of co-authors for this Viva la radioresistance article. And in this article, we actually provided a very clear idea. I will talk a bit about this a little bit later, that there's a lot of interconnections between space medicine and the challenges which astronauts will face in the space, and how this is all quite relevant for actual aging research and practical applications of, for human longevity on Earth. And yes, indeed, any humans which will go beyond magnetic shield of Earth, there will be a lot of known and also probably unknown threats, which we'll find out only once we'll face it. So I would say that there's quite significant probability that science and in particular space companies which are aiming to, for example, establish bases on Mars, they might really underestimate what will happen with astronauts once they will be traveling there and once they will arrive. And for this matter, in our opinion, also I totally concur with Arkady, before people will actually fly to the Mars, they should be significantly prepared with some reserves. So they should be protected from anything as much as possible. At the same time, if such technologies will be capable to protect them from such hazardous, aggressive environments, apparently in a normal environment, let's say, what does it mean normal? For example, any capital city today, it's not normal. Any big city, for example, such as London or New York, it's already an environment with quite significant level of different, chemical pollution or other like electromagnetic pollution. So there's a lot of issues in big towns, starting with, for example, that water and just environment, including food, in many cases, it's actually poisoned with heavy metals, unfortunately. So indeed, if astronauts will succeed with such advanced technologies, for example, such as genetic modification for them, with the aim to improve them from aggressive impact of radiation, application of such methods for humans on Earth definitely will protect them from like low dosages of negative hazardous impacts, including, for example, which many people experiencing living in big towns. Yes, of course, this study is famous, so it was mentioned literally in all previous presentations, but of course it's also in my presentation. So this is very, very strong case, because this here probably we shall pay specific attention on this multi-dimensional analysis, because in case of longevity, when we're talking about what technology, what set of technologies are required to actually extend healthy period of life of people on Earth, even in some kind of friendly environments, maybe even in blue zones or somewhere in in good places where there's no any kind of pollutions. But the point is that to understand what is required for that, we need to analyze human body really in super multidimensional framework and to apply multiple biomarkers. I would say hundreds at least, but better even thousands. And this is what Frank Curtis presented. So it's our priority to to focus on helping to further develop the ecosystem and sector of biomarkers of ethnic longevity, because this is crucial bottleneck and roadblock for further sustainable development of longevity industry. But the very same is applied for space medicine. However, the difference is substantial, because many scientists dealing with aging research, they have quite different mindset, literally psychological mindset, compared with scientists who are dealing with health of astronauts, because those scientists who are focused on protection of astronauts, they're very tied to actual human astronauts. Whereas, unfortunately, in case of aging research, many scientists tends to do too much theoretical science and spending too much efforts and time making maybe even successful, but nevertheless experiments on mice, instead of thinking how it could be and should be and ought to be translated to humans as soon as possible. And by the way, it is clear that in case of space, during many decades, they were developed safe and at the same time scientifically driven, sophisticated methods how to apply some different experiments, maybe with some micro dosages or with some kind of in a safe approach, but nevertheless to protect actually humans first of all. Whereas mice, it's just kind of could be considered a secondary additional experimental model, but everything is tied to humans. Here are some major takeaways from that article, Viva la radioresistance. At the same time, it's very much coincides. Let's say these conclusions were done back in 2017. Today is 2021, but I can, put my signature under each of these conclusions. So, first of all, we do think that not enough consideration were given regarding protection of astronauts if they will go beyond magnetic shield of Earth. In the article, we provided a roadmap how human, what are the ways how they could be enhanced for the purpose of radio resistance, especially for cases of deep space exploration and colonization, for example, colonization of Mars. And yes, we outlined several directions how this could be achieved actually, including, for example, how radioprotective mechanisms could be enhanced and how gene therapies could be applied. And we concluded that there's a lot of association between radio resistance and actual longevity, and articulated the position that enhancing human radio resistance is likely to extend the health span as well, either being applied for humans on Earth, or even most likely will extend health span of astronauts as well. Here are the links. You can review like all the materials which I mentioned in my presentation. You can review. You can get access by these links. It was back in 2015 when I announced $1 million prize for the first person who will celebrate his or her 123rd birthday, because the previous life record belongs to John Coleman, who died at the age of 122 years and 6 months. So since that time, Aging Analytics Agency and a number of other experts, they're actually, analyzing different practical longevity technologies, and we're applying those technologies to my own body because I myself, I'm going to celebrate my 124th birthday. And at the same time, I'm also going to fly to space in the next 2-3 years, maybe later to Moon. So that's why we are also starting to work on my body to prepare me for space flights, including Arkady Prokop actually also advising me on that matter. And there are a number of other specialists who actually helping me with to achieve that mission. So I would, as an investment fund, Deep Knowledge Ventures, which is deep tech AI focused investment fund, and Longevity Capital, which Kate Bass also mentioned. So we are very much committed, to proceed to keep a very deep attention into this interconnection between healthy longevity on Earth and space medicine, with the aim to enhance resilience of and immune system of astronauts. And I think during this year we will analyze much more deeply this particular sector. Whereas next year we will start quite actively to invest in this sector. Thank you very much for your attention.
Thanks so much, Dmitry. Stimulating and wide-ranging as always. It's now time for our panel discussion, so I'd like to welcome back all of our presenters and speakers to dive into some of these topics in a deeper and more dynamic manner. I suppose we'll start, since we're all semi-coincidentally co-authors of the 2017 radioresistance paper that's come up a few times. It's been some time since that came out, 4 years. So I'm wondering, given that the paper illustrated the broad spectrum of methods for enhancing the health of astronauts for deep space flight and colonization, and considering how long it's been since that paper has come out, I'm wondering, especially from Dr. Petut and Maheshti, how things have progressed, how well that paper is held up, if there's any specific approaches introduced in that paper that have progressed substantially since that time, as well as generally from all of our panelists, what their views are in terms of the short-term and long-term future of astronaut health optimization.
So I think looking to the paper in 2017, I think it was, and looking at what we are now. I will speak more at the level of the European Space Agency at the European side, and maybe Afshin can speak from the NASA side. But I think this information has been picked up by the European Space Agency, and you see a trend and a very strong move in terms of will to increase research in the field that we proposed in the paper. So as example, for example, regenerative medicine, we know that for long-term space exposure, space missions, we will have to think in terms of providing, if needed, like transplantation, bone marrow transplantation to have blood. So all the regenerative medicine has also changed radically since 2017. I think it's really boosted since then very much, and we are really going forward 3D bioprinting, but other ways also of organoid development in order to face this possible regenerative medicine that might be needed for the Moon and for Mars. So I see definitely a move forward, and that was also mentioned in the paper. Another move I see is hibernation and decreasing metabolism to increase radiation resistance. There also, maybe it's more developed at the level of NASA, but at the European level, we have a topical team, which is usually the first step to bring experts together to write a kind of a strategy roadmap for research. So we have that as well So I see, therefore, there also very much a move towards that. And then the biomarkers for radiation sensitivity of astronauts, for long-term exposure. This is really also pushed forward very much. And we also have at the European level a topical team on personalized medicine, because ESA really understand that we will need to go towards personalized medicine, that it comes from how a medicine is integrated in the body and works in the body. It's different in space, but it's different between people, between astronauts. So it's not only the pharmacology, but it's also the type of medicine to give. So there is really a big move. And I would say the paper, our paper, Vive la radiorésistance, is absolutely not out of date. On the contrary, I would say it was before, and yeah, very much in advance for its time. So this is my perception.
Yeah, I think I could jump in. Yeah, and I agree with Sarah. Yeah, I mean, I don't think the paper's out of date at all, because as we know, the field of space biology moves a little slower than, let's say, cancer biology, just because the experiments are hard to do. So we're limited with the experiments. There's only so much you can send up to space, and then of course the simulated experiments you do on Earth, like when I talked about like going to Brookhaven or some facilities, in Europe and so on. It's just limited experiments. So that there's still a lot of unknown that we covered in that paper that is pretty relevant. And I know, in my opinion, because I can't speak on behalf of NASA, but in my opinion, I know that NASA is focused on, for example, the 3D tissues and organoid models. They've been putting out a lot of solicitations based on that. So, to look at in the context of radiation models and how resistance, or, you could use those 3D kind of organoid models, or people who have them can use them to, develop and make it an easy way to figure out biomarkers to, countermeasures, in a sense, to come up with against it. I mean, in the bias part, one part I had microRNAs, which is one of my passions. That part, I've been actually diving into that a lot more. So that paper is still relevant in that section, because I've been able to start development on, which I didn't talk about today, but in a microRNA based countermeasure that could be both a biomarker for radioresistance or space health effects, and possibly a countermeasure that we start developing. So there's a lot. It's a good roadmap. There's a lot of things that need to be covered still in that paper that isn't known yet, but should be.
Well, that's great to hear, and I look forward to looking at that microRNA stuff when it gets published.
Oh, it was published back in November, the first part of it. So
great.
I can share.
So it seems, it's interesting to me that regenerative therapies seem to be a strong priority and a commonality between Europe and the USA. Would you say that the US is also prioritizing personalized preventive medicine in the way that Europe is, as Sarah mentioned?
Yeah, I mean, I think that's the goal of everyone. Just you want when you're astronauts in deep space, few thousand miles away from Earth, that you want it to have a self-sustained kind of system where you could regenerate the damage caused. So, one thing I know that NASA likes, which I've heard in the solicitations, is that that they like already repurposed drugs. Maybe off-patent repurposed drugs would be even better, because then you don't have to worry about a patent drug that's there. So in that sense, you can repurpose a regenerative drug already used for stem cell therapy or whatever you could think of. And if someone could think of how to repurpose that for space biology type research and effects of radioresistance, that's I know focus. That's a low hanging fruit for them if someone could do that. And I know there's a lot of solicitations out for people to apply for that, ask for screening, like doing those massive drug screenings on approved FDA drugs, and then see which ones will be applicable to what we're talking about today.
Right. And in your talk, you mentioned the fact that everybody knew about mitochondria, but the extent with which it was a central driver for a lot of these detrimental health effects, that is a bit of a new finding. Do either of you see that changing the landscape in terms of health optimization, mitochondria as a direct target for that, et cetera, in either the US or Europe?
Well, yeah, I mean, sorry, I'll jump in and I'll just briefly. Yeah, I mean, I could say once that paper came out, it got a lot of attention from the NASA folks, at HQ and other places. So, and I'm talking, it put it on the map for them. So that I think, so that's my opinion again. But, from the conversations I had, I think, they realized, okay, then this is a really important question, because we showed in a systemic way that, this is a true driver, it has to be. And there are, as we know, a lot of clinical therapeutics that are being developed for mitochondrial diseases or bioenergetic related diseases. So why not just take some of those and see if that works for us too, if in space biology. So I know that's changed a lot of their perspective too.
Yeah, well, it's an increasingly prominent focus within the longevity industry generally as well. So, so that's interesting to hear.
I think to me, I think the mitochondria is and the rest of the genome. We've got to also tackle a lot the DNA, the mitochondrial DNA, and we know for radiation response that you have a specific mitochondrial DNA repair system, and in comparison with the rest of the genome. So there are two different kind of world, and we start to know more, but there is still a lot, I think, very much to learn from how do mitochondria interfere, play a role in the radiation resistance, and still a lot to work. And it's not, we understand now, it's not only the nucleus we have to focus on, and the DNA and the genome from the nucleus, but the DNA from mitochondria is certainly extremely important, certainly for radiation protection purposes.
Yeah, 100%. Yeah, there's more unknowns than knowns, I guess. That's really, when it comes to space biology and what's affected.
Absolutely.
So plenty to work for, keep us in business.
Yes.
Absolutely. Well, we do have some questions from the audience, so I'll read a few of them throughout this panel session. So Sarkis Hiritsan is asking, is it possible to create a new human species capable of enduring deep space detrimental environment? So new human species, I know we mentioned, genetic modification. And I also will combine this question as a follow-up with mine, which is whether anyone is aware of any space projects that specifically focus on human reproduction, whether modified or not.
But there are some space projects on human reproduction, but we know that there is really there are a lot of difficulties in terms of reproduction. And a lot of studies have been performed on fish. Some experiments from the Russian side on dogs, but all in all, that's not so much on really embryogenesis, on spermatogenesis, but there is a need to do that. So again, I will speak from an ESA side, from the European Space Agency side. We are doing bit like NASA, every certain number of years, we do the scientific roadmap and the strategy. We define the strategic roadmap for a certain number of years, for 5 years. And what is really interesting is that for the first time this year, we have a dedicated roadmap for the reproductive system. So there is really also a trend on tackling the reproductive system, whilst before it was not considered as a priority. So now it's becoming a priority, because of course, if we want to think about staying on the moon and maybe colonizing later on other planets, it needs embryology, and the development needs to be working well. But there are really a lot of constraints and a lot of difficulties, because we know that there is a lot of, I've got a lot of echo in my microphone now. So there are a lot of constraints. The first stages of development are impaired by space conditions. So if you do some in vitro studies, the very early stages of development, from the fertilization to the end of the pre-implantation period, there you have the migration of the embryo along the fallopian trunks and then going down in the uterus. So these first stages, usually are quite similar in many mammals. So you can use the studies that you perform on mammals to get some ideas on what could happen in humans. And you see quite strong differences. So when you don't have gravitational force, when you induce some impact. We also know that from studies performed by NASA scientists from birth on in space or just after birth, so perinatal period, just after birth, a few days after birth, that if you expose young pups to weightlessness, they don't develop so well, and in particular the hind limb. So the muscles from the legs are not developing so well, because you need this genetic stimulation and in order for the muscles to develop. So there are really quite a lot of constraints, also hormonal constraints, a lot of changes, hormonal changes in men and women in space. When you look at sexual hormones, quite a lot of impact there. So from data that we know from the Apollo missions, where everything was published, thereafter there was a lot more concern about protection of private life. But at the level, at the time of the Apollo missions, there were quite a few studies on libido and hormonal levels and so on, and there it was very clear that libido decreases, so that sexual activity kind of decrease. So microgravity has got an impact seem to have an impact on that as well. So but I see that there is also more and more interest now to understand that better and to study that better.
Well, it's really great to hear that you're seeing it as an emerging research priority, given that the future of space colonization quite literally depends on healthy reproductive ability. Afshin, anything? Are you seeing similar tendencies in the U.S.?
I mean, well, one thing you mentioned, yeah, I mean, as NASA has a decadal survey every 10 years, and that's up now. So, that's usually welcomes anyone from the community to put in a white paper. If anyone here can do it. So, if anyone's interested, check it out. Type in NASA decadal survey and you'll find it, the call for people. But the last 10 years they weren't focusing on that. So I don't know if this new decadal survey, if people like you guys here submit a white paper on, we should focus on that, or NASA should focus on that, they might start paying attention to it more and more, because I'm sure there'll be people asking for it. So yeah, and as Sarah said, there've been some studies that actually, there's one data set on GeneLab that looked at the rat pregnancy and like lactating and things like that, and find, so that data set is actually available, a transcriptomic RNA sequence, or no, it's microarray data set that's on there. I know JAX has done some studies on, the Japanese Aerospace Agency. They've done some studies of the embryonic type cells, like from mice they've sent up to space and then brought them back down to see what happens, after trying to get the mice to say implant them back into mice in the reproductive organ, and they've seen some interesting effects too that might even just like the embryonic type cells, and they do the fertilization afterwards, they see some impairments that happen. So, but yeah, I'm not too aware of too many studies around the US other than what Sarah mentioned.
There is a study that's started right now. I think that it flew on Thursday last week to the ISS, and it's a study on mini octopus. And it's not it's not reproduction, but they are very, very young mini octopus. They are 3 millimeters long, and they are not colonized yet with bacteria, so they don't have any bacteria in their gut. And the idea is to understand, because that also might be a very important point, is how bacteria, we are born kind of sterile without bacteria. So how these bacteria are going to colonize. These bacteria are fluorescent, and you have two little aquarium, an aquarium with this 128 mini octopus, and then a little aquarium next to it with these bacteria, and you open these the door between the two aquariums, and you let the bacteria move to the mini octopus and start to colonize the different parts of the body. And it's very interesting to see also, because it can the immune system of the mini octopus is quite you can draw some parallelism with the immune system of humans. And And also from newborn stage, how kind of the colonization of bacteria is taking place in an organisms like the mini octopus. So I think we can draw also some conclusions and some interesting conclusion on how the immune system starts to develop and how this gut microbiome is starting to develop very early after birth. So these are also, yeah, it's an interesting experiment that I wanted to mention because it's quite spectacular when you look at this little mini octopus, how they look like.
I'm very glad that you did mention it because it sort of presaged my next question, which is going to be about microbiomes as another emerging research focus, which is lucky considering the bacterial origins of mitochondria, so it's topical. But I was surprised to see it in the presentation today because we have spoken, and I know that Dimitri has spoken on this to some length, at least privately to me, about the role of mitochondria in practical longevity and things like that. So besides that octopus study, to what extent is the microbiome becoming an emerging focus? I would imagine that it hasn't been for too long, and I was a bit surprised to see it up there, though glad to see it there as well.
And Frank,
for me, yeah, it's a very important and hot topic. Microbiome is emerging and going full speed. I think from all sides. You see that on different types of disease and how the microbiome is becoming dysfunctional. We have one publication that came out today in Nature about dysbiosis after radiotherapy in a mouse model. So it's really important to understand how to keep the right balanced microbiome, because yeah, this is I think it's very crucial. And there I think NASA is much more forward than ESA. NASA is implementing research and doing is much further than the European side. So we have it in our scientific roadmap and strategic roadmap at the level of ESA. I'm very happy that we have it since this year. It was not in the last 5 years. It was not in the roadmap, so it was not in the strategy. So I'm very happy it's now in the strategy, but it's a booming field.
Great. And guys, I believe that Arkady raised his hand and has a comment as well. So Arkady.
Yes, I would like to comment regarding genetic modification, as the question was. Yeah, we need to understand that nature continuously plays with genetic modification. Millions and millions of years ago, our ancestors continuously underwent mutations, and though they originated in the tropic areas of Africa, they conquered north, they conquered polar regions because of mitochondrial mutations. Yeah, because the possibility to produce more heat instead of more energy as it needed. For instance, you need to run from the lion and you don't need that much heat, but you live in a polar region, you need to keep your body warm. That is the difference between Kenyan marathon runners and Eskimo. The same is happening now continuously. As I already told in my talk, that we have a plethora of mitochondrial diseases that are incurable currently, and no smallest possibility that we can do some pharmacological intervention to improve quality of life of lay patients, like the boy which I showed. But the boy, if he will stay in an environment with 11% of oxygen, he will continue developing quite normally. But it's extremely difficult to provide such environment on the Earth. But in space, no problem. So I think that can be an interesting possibility for people who have these mitochondrial diseases, incurable mitochondrial diseases, if we create new environment which supports their development and supports quality of life, that can be very helpful for planetary explorations. That's my opinion.
Absolutely. Thank you, Arkady. We do have another question from the audience. So on longevity side, we can look at supercentenarians for clues on age resistance. What are the equivalents, if any, for humans who are radio resistant? So age resistance, radio resistance.
Sarah or Afshin, do you have anything to say on that matter in terms of,
Any parallels?
Yeah, I mean, I guess for space related radiation, I mean, we know people do experiments with, the different strains of mice that are, as I mentioned, like the C57 or more radio resistant than the BALB-C mice, which are radio sensitive. So people have seen parallels there, definitely when the C57 mice, of course, have better survival with space radiation experiments to see when you do simulated ones and, at the Brookhaven National Lab. But I mean, I've done some aging work related with cancer risk and space radiation effects, and definitely there's, when I did that work a while back, what we did see is that that with increased age, it seemed like you became more radio resistant, or you had the like for angiogenesis models, or the tumor growth in this case, became the tumors didn't grow as well. Well, in general, tumors don't grow as well when you get older and older, like when you become, like 80 years old equivalent human for a mouse, for example, it doesn't grow as well. But, when you add the space radiation, like a proton radiation or iron radiation on top of that, you see that, optimally, the tumor doesn't progress as well compared to a younger person, which could say maybe we should send 80-year-olds to space because they're, for cancer risk, at least. For the other risks, I don't know about.
Yeah, this is, by the way, this is a very interesting point, because Sarah also showed such graph in her presentation, that risk of cancer actually decreasing with age, and that we shall send to Mars people who are older than 50. So, I mean, why it is happening at all? I have just one idea that with the age, the telomerase length is actually shortening. Maybe that is protection mechanism.
Yeah, I mean, that study is done by Susan Bailey, and is the one who's really, I'm sure you guys seen all her studies that looking at the telomerase shortening. It's an interesting study because when they come back to Earth, most of the times the telomere is recovered in length.
Mm-hmm.
So it's as she would say, it's like the hypothesis of are you growing younger? They don't say, because it's a tough one to actually say if you grow younger in space. And of course, the media would jump on that, say you go to space, you become younger, and that's not really true yet. But the telomere length is definitely something's changing with it. And how does that affect your radio resistance and age? Let's say aging in space. That more research should be done, but it's an interesting one to look at.
Yeah, and if I may comment on what Dimitri just said, and what I've seen. So we see and end of next week is a very important date for all the space motivated people to apply for the next ESA astronaut campaign. So we all the ones who would like to apply have to apply before the 18th of May. So I will apply, and what is very interesting is that in Europe we have this kind of application procedure once every 12 years to 15 years, so it's not very often. And this the last campaign, the limit of age to apply was 37 years old, and for this campaign the age limit is 50 years old. So it's been taken up by ESA for long-term flight to the moon and to Mars, to take on board people who are slightly older. So that's picked up as information.
On Earth, now there's more and more recognition about such problem as such kind of sociological problem as ageism. So now there's a lot of initiatives to protect all people from discrimination in rights. For example, we do have one project. It is named Longevity Banking Card. We invested in the project. They're going to hire. So that project will deliver fintech solutions for all people. Like people older than 60, they're targeting people for 70, 80, 90. The idea is they're going to hire as testers of those services people who are older than 80. So my point is that, I mean, if to extend this, course of thinking, we probably, because now it's also becoming, normal to consider that life expectancy on Earth will be beyond 100 years and probably even 120 years. So my point is that probably soon we will see maybe tourist astronauts who will be significantly older. Maybe they will not go to moon or for long flights, but maybe, you know, just for kind of maybe for one day. But I wouldn't surprise that in the next 3 to 5 years we'll see 80, 90 years astronauts.
And speaking of supercentenarians, because that issue was raised, it so happens that Age Analytics Agency has another open access report precisely devoted to the database of supercentenarians or super old, which is anyone who is 110 years or older. So Dmitry mentioned that he established a $1 million prize for the person who reaches 123 years. So we're actually closely moderating the database because there are some who are approaching the threshold. And in terms of you know, the publicly available profiles of supercentenarians and perhaps making some conclusions, you know, you can take a look. There's an interactive database and you can try to figure out what played a role.
So and I'm not sure what kind of samples or data you might collect from these folks, but I could imagine like an ultimate experiment to design, you know, if you get proper approval from IRBs and everything to do is get samples and then irradiate their cells, for example, in front of Brookhaven National Lab beamlines, or do those microgravity experiments, or even send those cells in space, you know, because you could send payloads. So that's something to think about if, like, you know, from your folks that are involved with your studies here, if they're willing to donate samples to space biology research, you know, that I think we'll get to the question that was just asked here about, you know, age resistance and radiation resistance.
Excellent.
Yeah, well, you read my mind, Ashim, because I was going to, you know, remark on that. Yeah, a supercentenarian radio resistance study.
I think we have several questions also to Arkady, but my particular question, because Arkady was mentioned this particular point in his presentation, and this is quite interesting because I think there's a lot of kind of like say like addiction, marketing addiction towards antioxidants. So my question to Arkady, and I probably will conclude with several people who are asking this in the chat, whether antioxidants, they're actually beneficial, placebo, maybe they're negative, and what are your prediction in terms of if I'm going to space, what may be some, you know, medical supplements which are probably beneficial on Earth for health longevity, whether they are also going to be beneficial if I'm going to fly, for example, to the moon, including such as antioxidants, if they're at all working.
Yeah, thank you, Dmitry. That's a very interesting and very important question. From all what we learned from 30 plus years of studies of antioxidants and working with antioxidants, it's very easy to overdose them because most important antioxidants we become not from the food, not from the exogenous components. Most important antioxidants are our antioxidative enzymes, which are produced in the cells and mitochondria. And these are superoxide dismutase, catalase, peroxidase, and also glutathione also comes to this, yeah? But if we use excessively exogenous antioxidants, we suppress production of endogenous antioxidants. And this immediately shifts the balance of regulation of redox regulation of the cells. Cells continuously undergo waves of oxygen and oxygen radicals oscillations. And these are extremely important signaling functions. They also participate in immunity. They participate in synthesis of hormones, in neuromediators, and just putting a lot of antioxidants above it. I sometimes compare it with, you know, you have a wooden house, you have an iron stove, and you need to cook on this stove, you need this stove to make your house warm, but you continuously put water on it that you are afraid it will burn too strong and it will damage your house. That's exactly what is going on with excessive antioxidative supplementation. I had many patients who damage their health immensely by continuously taking antioxidants for years and years. So I think in space, it will be just very informal people who will use these approaches. They should be very quite informed, and they should have some individual recommendations when and how to use antioxidants.
If I may bounce on that, I think the idea of using antioxidants is mainly for extravehicular activities, to maybe take that boost of antioxidants before you go out of the space ship or the space station or the moon station before you would go out. So that would be really on the case and for specific activities. But this is how I would see it, but not on a prolonged way, but specifically.
Yes, I do agree. Just taking them in a preventative mode, but
Yeah.
for shortened time, for special periods. Yeah.
Yeah.
But from all what I know, from what studies I also did some studies in this aspect. Yeah, the hypoxia, 10% oxygen instead of 21%, had extremely powerful antioxidative effect.
Mhm.
Especially after many sessions, after many preparations, antioxidative intracellular potential raises immensely.
Yeah, I
And
agree.
it also increases radio resistance, resistance to some toxic substances, and it increases, for instance, in the neuron, we see increase of neuroglobin. We have hemoglobin, we have myoglobin, but in neurons, we don't have any oxygen reserve, oxygen bottle. But if we undergo the intermittent hypoxic training, each neuron produces his own oxygen bottle, neuroglobin. And this explains a lot about the protective action of intermittent hypoxic training.
That's interesting, Arkady. To circle back briefly on a point you made earlier, you were mentioning how space travel is ideally suited for health protecting interventions that are mainly environmental and coming in from the outside, as opposed to direct biological therapies and things like that. Because space flight needs controlled environments in the first place. That line of research seems to make a lot of sense. I believe was a point that you were trying to make earlier. I'm wondering if Akim and Sarah see any tendencies or convergences there in terms of environmental control approaches to health span extension or the protection of the health of astronauts.
Go ahead.
I will be short. To my opinion, expansion of healthy lifespan in our environment at sea level is not optimal. It will not work optimally. The effect will be much better for if we, let's say, if we're residents of middle altitude, so about 2.5 kilometers to 3,000 meters. Yeah. At this altitude, oxygen partial pressure is lower, and all the metabolic oxidative pathways, they are functioning just better. There is a huge statistic. Millions of people live on this altitude. Yeah, let's say even till 4,000 meters. And statistical research, epidemiological research says that they are better protected from cardiovascular disease. They have better protection from cancer, from malignant diseases. In spite of that they're also drinking, smoking. Nevertheless, they have statistically better health and have better health, longer health span. So I think that in the artificial environment will make a lot for extending healthy longevity. And this environment is very hard to create on the sea level. But either you should live in the mountains or you can live on board on space flight, on the spaceship. By the way, from private communications from our cosmonauts, that was about 12 years ago, I asked them, do they feel comfortable at 21% oxygen? And they say no, because they have, this problem with hemodynamics. They have always some headaches and problems with sleep. So they tried different approach. They tried to reduce oxygen content till 18%, and everything was improved. I don't know. I'm not sure if they shared it with the support team, but that fact that they did it, I remember perfectly well that they were very happy with this experiment.
Ashim and Sarah, any commentary with regards to
I would say that aging and radiation effects have got a lot in common. So if I look at the way skin reacts in space and skin age also more when subjected to radiation, or the immune system or the bones. So there are few organs that are really aging much faster in space conditions, and I think we can understand a lot, and it can help a lot to understand how space interacts and in this accelerated aging, in order to help on Earth to better mitigate that for different diseases and against aging and to increase longevity. So I think there is really a lot. But also at the level of the molecular mechanisms, not necessarily only on studies on astronauts, but a lot on, I think, on platforms like the random positioning machine, which is a well-known platform to perform in vitro experiments. And other platform like the hind limb model that was presented. And so I think there are models also on Earth that can be useful, that are space related models that can be useful to understand aging and to develop countermeasures for increasing longevity. So space can really have an important contribution to improving our understanding on accelerated aging and increasing longevity. So I think there is really a lot that can be brought from space research to the longevity field.
Yeah, for the environmental factors, I think I agree with our Katie that I think it's hard. I don't think there's any really. I mean, I guess on the ISS we do know that there's related maybe to the oxygen levels. We had to put out a paper a few years ago where we shown that that definitely there's there's about 100 times more CO2 levels in certain regions of the ISS, like because it's a closed tube. So let's say the average CO2 level on Earth is what, 500 ppms, parts per million, and someplace in the ISS it's like 5,000. So it goes really high. And I know Scott Kelly has reported like exactly what the cosmonaut said. He gets headaches sometimes in certain areas of the ISS and the oxygen levels. So I don't know what they did to mitigate that, but they probably should have listened to the cosmonauts and increased so Katie says, increase the levels of oxygen. They might have been okay. So I mean, things like that environmentally they could use to improve conditions. But other than that, like I know the Japanese are doing some experiments to the JAXA side where there's artificially simulating gravity through their cages. So they have a little system, kind of a centrifuge system where they have mice in there and they artificially induce a 1G environment in the ISS. So that's another way maybe for the environment to artificially simulate something that could be more beneficial and improve age, improve your mitochondrial bioenergetics and things like that right now you can't do. But I mean, I guess other than that, like there's nothing, I can't think of anything that would mitigate space radiation effects from your environment unless you encapsulate a ship with like enough water outside of it as a shielding, as we all know that do that, but then the ship will never leave Earth. That's, everyone knows that probably at that point. But yeah, that's a tough question to tackle, as our Katie said too.
Well, a specific question from the audience with respect to cosmonaut or astronaut protection. So John Ferber is asking, could the panel comment on the radiation protections of humans in an O'Neill L5 colony versus a Moon or Mars base? the O'Neill colony would have still been made of asteroid material as well as centrifugal gravity to maintain muscle tone. So it's quite specific. So I wonder if anyone can comment.
Not too familiar with the O'Neill system, so.
No, but I guess that's if you have a different gravitational force on the moon, on Mars, or the O'Neill, that will impact slightly differently the body. So, but we don't know exactly how it will. We expect that there will be some differences, but it's difficult to predict already now. If on the there are different hypotheses saying you need a little bit of gravitational force to stimulate everything to work back almost normal. But yeah, is it like that for every part of the body, or are each part of the body or each organ reacting differently? It's difficult to know at this stage. But
I know
don't
for that JAXA experiment that they simulated, maybe something similar as O'Neill thing, where we tried to simulate the 1G in space, which did pretty well. The muscle functions were improved a little bit from their experiments. I have to go back and remember how well it improved that. And I guess there's Seward Rudkov at Beth Israel, a collaborator of mine at Harvard. He has that partial weight bearing rat model where he does the different percentages of gravity, on Earth simulated. And you definitely do notice different muscle functions, improving that way. But again, that's simulated on Earth through a rat model, not really what's happening in space in this O'Neill model. But you can get sort of an idea of, let's say, the gravity percentage on the moon compared to, let's say, Mars simulated, and then doing like the really high limb, which is deep space, the true microgravity experiments. So you do see differences in muscle function and mitochondrial function and other factors that happen.
We see we in our random positioning machines we can simulate Martian gravitational force, lunar, and then full weightlessness, and we started to do different studies, and indeed the response seems to be slightly different, but it's a bit too early now to say. Yeah, it's very how different it is, but I predict it could make an impact. But I mean, the body is composed of different organs, and they work together in systems, but they also work autonomously, and they have each a different response, a different a tolerance to space conditions, and so that also makes this, the whole story a little bit complex, on top of the fact that humans react differently. There are differences between people, gender differences, age differences. So it's really complex. But I think technology and technological advances very much help to decipher this complexity and to understand that better.
Thank you.
Circling back a moment to our earlier discussions on how Europe's approach versus the US's approach may differ, et cetera. Sarah, I know that you're a member of the United Nations Scientific Committee on the Effects of Atomic Radiation and the High Council for Health. I'm wondering if you and Ashu see any differences in how large multinational committees are looking towards these things, particularly with regards to some of the therapeutic modalities in our 2017 paper, which at the time were perhaps a bit futuristic, but which are now being onboarded by various national or at least continental agencies. Do you see stark differences in terms of the future facingness, for lack of a better term, for committees like the UN committee that you're on?
From the UN, it's very interesting to see the differences between countries and in the way they tackle the radiation problematic, the way. Yeah, if I take some examples of Chernobyl or Fukushima, it's so UNSCARE from the United Nations is reviewing the reports that are provided and comment. So each nation has got a possibility to comment and to approve or reject some sentences. But there I see some strong differences sometimes between countries. Now, in terms of therapies, I would say it's not so much. I think Western countries, are trying to develop new ways of treating. but it's quite similar, I would say, in all the Western countries. I would say it's more depending on the type of hospital and it's more inside the country itself. It's not that much different from the different Western countries, if I may say. But I was really impressed by your presentation, Franco and Kate, because I'm thinking, yeah, at the level of Europe, there is so much to do. When you look at, it was very clear the difference between the US and Europe in terms of the femtech but also on terms of biotechnological development. Yeah, I would have expected that Europe would be further, but I was a bit disappointed by that, and I think there is still a way to go. But on the other hand, it was very inspiring to see that, because there is
Thank you, Sarah. Well, it was very inspiring to moderate this very robust panel. We actually have quite a few more questions from the audience, but unfortunately we don't have the time. So perhaps it's an indication for us to have a new conference, an event in a few weeks or months' time. It has been our great pleasure to have you over. Again, it's exciting conversation and audience agrees. Thank you very much for your time. We also thank all of our attendees for many questions, for hand raises and all that. I think we should be concluding our day 2 of our conference. We've mentioned earlier that this is a recorded session, which will be distributed thereafter.
Thank you very much.
Yes. As Kate said, it's time to wrap up, even though, just based on the nature of this discussion, I'm sure we could have gone on for another 3 hours. So yes, thank you once again, Drs. Batut and Baheshti, for joining us today, as well as Arkady, Dimitri. Very stimulating discussions. We will be following up with all of our attendees with a video recording of today's sessions, so watch out for that. I would also encourage everybody to log on to aginganalytics.com and subscribe via the subscribe button near the bottom of the page in order to stay tuned and up to date with regards to future project and events announcements. As I mentioned during mine and Kate's opening, we are in fact planning another several physical and virtual events on the topic of practical applications in mitochondria research for human longevity, space medicine, and a number of other topics. Looking at London this October, and something in the US during the first few months of 2022. So please keep an eye out for that. And thank you once again, one and all, speakers and attendees, for joining us today. And we hope to see you again soon.
Yes, thank you very much. And I would also like to add a special thank you for co-organizers of day 1, Kinexium and Kitalis Institute. We had another robust discussion yesterday, so we hope to have more events in the future together. And our view on longevity is rather similar, and we encourage attendees who are interested in the topic of longevity to consider visiting the virtual conference of Metabicity in Washington, D.C. It's a virtual one, so you can go to www.metabicity2021.com. And the link that Franco mentioned, aginganalytics.com, in addition to just staying informed about our future events, you can actually download all of the reports that we've mentioned and a lot more. So this is open access, and I'm sure that it will be quite educational for the audience. Thank you.
Indeed. Thank you, Kate.
Thanks,
Thank you.
Kate.
Bye, everyone.